Okay, a very good afternoon to all of you. On behalf of Department of Bioinformatics, University of North Bengal, I, Dr. Shubharna Thakur, welcome you all for this one-day national webinar on career opportunities in bioinformatics. Now, bioinformatics is a very emerging interdisciplinary field of science that focus on developing methods and software for analysis of biological data. And over the last 30-40 years, breakthrough in molecular biology and computer science have combined to establish the area of bioinformatics. So this rapidly growing field of science has huge job prospects in all sectors of biotechnology, pharmaceutical sciences, agriculture sciences, biomedical, or even data sciences. So this webinar is a great opportunity for the young undergraduate and postgraduate student to understand the career scope of bioinformatics and the related fields. So we are privileged to have with us two eminent industry experts with vast experience who will explain to you the opportunities in this field. They will also talk about the qualifications and skills needed to get hired in this industry. So I'm sure this webinar will help you immensely to plan and shape up your career in this field. For the inaugural ceremony, we are honored to have with us our Honorable Dean, Faculty of Science, University of North Bengal, Professor Subhash Chandra Roy as our chief guest. So we are joined by Dr. Amit Das. Before introduction of our expert, I just want to give few instruction to the participants. So please, all of you, keep your mics off. And if you have any question or query, you can post them in the chat box. So chat box is there, so you can post your query in the chat box, and at the end of this session our expert will take up your queries. And another thing is that at the end of the two technical sessions that we're gonna have, you will be provided a feedback form. So the link will be shared in the chat box, so you have to fill up the feedback form, and then based on that you will be delivered the e-certificate. So all of you are requested to check your the chat box at the end of the technical session too. So moving on with the first technical session, so we are very privileged to have with us Dr. Amit Das. So I'd like to introduce Dr. Das to our participants. Dr. Amit Das is presently working as a senior data scientist in Deloitte, India. He has more than 6 years of industrial experience across healthcare, biotech, pharma and data analytics. Dr. Das specializes in different aspects of data sciences, including data automation, assessment, cleaning, analysis, visualization, and data-driven decision making. Academically, Dr. Das graduated in zoology from Presidency College, Calcutta, followed by Masters in Biophysics and Molecular Biology from Calcutta University in 2009. He obtained his PhD in Bioinformatics from University of Kalyani in 2016. Dr. Das enjoys mentoring young minds to prepare them for their industrial career and subsequent growth. He is also passionate about increasing the penetration of digital tools and techniques in day-to-day life safely and ethically. So Dr. Das, the dais is yours, so you can join and give your lecture.
So, as Dr. Thakur said, that you already know that I actually started as an experimental cell biologist. Just a minute. As an experimental data scientist, so I was doing this cell culture and all. Then during my PhD from 2013 to 2016, I actually started doing bioinformatics, and that's when I started coding. So I knew programming like from my very early days in school, like high school, but not actively doing. Mostly active coding I started 6 years back or something. So you all can be assured that since you are a bioinformatics student, you are quite a bit have some experience in doing programming. So the transition from bioinformatician to a data scientist is not actually that hard or not that big journey. It can be done fairly easily. So we'll discuss on those. And then I have like a 4 years or so. I have been to my first company, then the two other companies. Right now, I work for Deloitte. So for some regulatory reasons, I am not authorized to directly always use Deloitte. So I just say like I work in the healthcare domain. So I basically in the Deloitte, although it's known for a service company, the management consulting company, they also have a product in healthcare. We'll be discussing those things, so what I do and what those kind of things are, how I will be doing. So basically a decade-long journey I have from transitioning from a cell biologist to a data scientist. Yep. So what is data science? I think these days AI, artificial intelligence, machine learning, deep learning, Internet of Things, or IoT, data analytics, these things are like everybody, all of us heard about or something or somewhere else, things. And then people obviously talks about, okay, if you are doing data science, you need to be know maths, stats, advanced computing, you need to be like a geeky hacker and all this. You need to be a data engineer, you need to have a lot of scientific mindset put into place. So, Dr. John, just one point. If in between there is any trouble, just directly unmute yourself and let me know.
Sure. Yeah, okay, sure. Yeah, okay.
So all these are like people these days associate with skill, and whomsoever he's called like there is a very popular synonym that like a data science is so-called the sexiest job of the 20th century. Many of you might have heard about this. There has been a number of talks about those things. Honestly, these all are data science, but this does not at all make sense when you call about what is the impact of data science. So basically, the impact of data science can actually be summarized in one single slide. It's all about providing data-driven insights for decision making. So whatever you use in the background, it doesn't matter. Whether you simply do the calculations in pen or paper, you do the calculations in like Excel, or you use like R or Python or even the high-profile deep learning libraries. That's all like your day job or your bench work that you are doing. The only problem, there is a business problem. You have been provided with some data, or you have gathered some data, you do the analysis, and you provide a solution or some insights supported by the data. So that's all is about actually the data science. So data science is all about data-driven insights. And I would actually share one of my experience is that. So when I was joining my second company, I asked my potential manager that, what do you want from me as a data scientist? And he was not a data scientist. He was actually a research scientist and almost 59 years old. But actually his answer gave me, made me right at my point that I am going to accept this job. What he said is that, I want you to help us in exactly the same thing, provide some support, whatever you do that's your, I don't understand those things, in the decisions that we make. And why it was such an important for me to take the decision that, okay, this person could be a good manager, like a good teacher, a good mentor, is that many times if you read news about data science or companies implementing data science in their organization, people will say that we have now this year investing 5 million, 10 million, $100 million, and we are trying to achieve a 5%, 7% or 10% growth, and we will be hiring 50, 20, 100 data scientists. When people make up this mind, or when people have these assumptions about the numbers that they want to achieve, that's like a red flag, that's like a roadblock. How that person actually knows that they're going to achieve 5% growth by hiring 50 data scientists and spending $50 million? Is it supported by certain data, or is it just like the management decisions and the assumptions that they are going to achieve? Goals are always good. But if the goals are very far-fetched, like somebody say, I want to publish a paper in Nature in the first 2 years of your PhD, yeah, that can be a good goal, but that can actually be stressful as well. So because a PhD student goes through a lot of training, and that may not be achievable in the first 2 years, it might be achievable in the 5th year. So similarly, when people need to be actually very maturely handle what data science is and what data science can do. It's not like a magic trick that it can give all the solutions. If there is no data, there can actually be no insights. And honestly, more than half of the industry is actually struggling with having access to data. Historically, a lot of data has been there, but many of them are so unstructured, or like they have been written like a paper written, they're not digital, or they're like a human readable only. So those are not actually useful. So data science is a hype, but it also has quite a good benefit that we will discuss. So why is data science actually being used? Data science in these days is almost used everywhere. Many of like the students who are here, you might be using Facebook or Instagram. And you might have seen that when you search for something, let's say you search for a beauty product in Google, uh, instantly in your Instagram feed, the companies who sell that product comes up. Or if you search for a computer or like a phone, immediately phone related ads comes up showing up in your Facebook feed. So how these are happening? It's all because of your search data, historical data that are being tracked. So these things are actually quite known to you. So I will not go much into on all those details. Some of the uncommon one that probably industry is very well versed with, but it's not that well clear to you. So that I will be discussing. So social media marketing and the marketing part we just said, like that's for like a updating your feed. So one of the other definitely self-driving cars you have seen, it's purely based on image analytics. So the car actually has a few cameras, front, back, side. So each time it is feeding numbers of images and capturing, and those images are actually being processed real time. And based on that, it is actually making some decisions, like how the distance from the front car to the this present car, and then accelerating or decelerating accordingly. Pilots and the drones use a lot of imaging data, satellite data, weather data to actually navigate the path. And you will be surprised to know that weather data actually plays a lot of role. So there are few incidents like when the long range flight, let's say US to France flight or so. So those are delayed, but they use the weather data and they use the jet stream to actually propel the flight faster and cover up the delays. Like we train, we know, right? That the trains cover up some of the delays when at night when the tracks are clear. So pilots also use a lot of data that are being made available to them and use it efficiently. So as more data is being fed to the pilots in a use. Healthcare is a big time use of like a user of data science. I am right now in the healthcare domain. So one of the field is like the supply chain analytics. So supply chain analytics, you might know that during this COVID time, initial supply of like paracetamol, then the malaria drug, and then many other drugs during COVID. It came like a crisis of the crunch. So did we not have the production? Yes. Initially, we did not have that level of idea that the production need to scale up that level. But when it became clear, there was an initial hiccup, but within a few days, let's say 15 days or 2-3 weeks, all the supply chains ramped up accordingly, or they are optimized in such a way that all the COVID-related medicines could actually be provided as fast as possible to the zones required. And data science also played like a how much will be the distribution. So let's say if there is a cases are hike in a particular state, whereas compared to some other state, then it doesn't take a lot of mind to think that we need to divert more medicines or the demand is high. So these simple things can actually also be used in healthcare. Healthcare, there is also a number of other things being used, including like the disease prospect. So like the how is going based on the, even if you take the COVID example, based on the number of hospitalization, number of oxygen requirement, all these things, so the hospital are actually real time predicting how is the demand is going to be in another 3 hours, and based on that automatically they used to place oxygen demand or something, that okay, in 6 hours we are going to need this much liters of extra oxygen, if can anyone provide. So this is how real time tracking actually helps in healthcare. Sales, we all know how Flipkart, Amazon, they all use our data and analyze it to like a demand forecasting. So we all know this big billion days, right? So, but many of us have not actually faced the problem that okay, we ordered something in big billion day and actually had to wait for 10 days to come. Most of the time it comes in 2 days, 3 days, and now they are making 1 day. But this demand is like a peak demand when this 7 days, 10 days even goes on. After 15 days, those demand will not be there. So they need to optimize the supply chain or like the endpoint delivery system in such a way that they are ready for when the demand starts, but they also can scale back when the demands decrease. So Amazon has these Black Friday deals in US or the Christmas sales, Thanksgiving sales. So they use immensely for like demand forecasting. One of the interesting things that I can tell you, like the use of weather data. So the Nordic countries or the most of the northern European countries, they hugely suffer from snowstorms or snowfalls. So most of the morning flights actually need to be cleared by using a particular fluid that actually removes the snow from it and makes it a little bit lubricant, so that when the flight takes off or on the fly, the snow does not stick off to the wings, because then the aerodynamics goes away. Earlier, airports has to massively store those liquids and those liquids also become costly, and their supply chain is also equally costly. But if there is no snowstorm for 3 days, then they have actually paid X price that they could have used for some other purpose 3 days for 3 days or something. So using actually weather data, they are predicting when snowfall is coming, and based on that, they are optimizing when to order that particular fluid for de-icing, and then also actually they scale up or scale down based on the demand. So weather data is playing a lot of role these days, and also with agriculture these days, because nobody expected to have a hailstorm in Sydney. Sydney's never had actually a hailstorm, but last year I think they had a hailstorm. So now they are trying to predict: are we going to have those kind of events in future as well? And those huge hailstorms actually cause a lot of property damages, which goes to the claim to the insurances. So insurances are buying health weather data and trying to see how they can optimize the insurance benefit and the insurance's premium based on the forecasted events. Yep. So a lot of areas, almost everywhere these days, data science is being used. So how actually data science role looks like? What does a data scientist really do, and what are the different roles that they need to play? As you can see, like I have mentioned, like there are different roles. Broadly, there are 4 categories. One is data scientist, there is data analyst, then there is data engineer, and there is a data steward. So in general, there is a lot of also overlap between these roles. So a data scientist cannot be a data scientist without knowing part of these 3, or a data engineer cannot be a data engineer without knowing the other 3. But there is a certain level of expertise. So let's say you cannot be a molecular biologist without knowing a little bit of cell biology or how physiology works, those kind of things. But your expertise might actually be in molecular biology. So how these roles are different? So data analysts spend majority of the time bringing in insights out of the data, whereas data scientists play majority of the role in innovating different methodologies to analyze the data, or exploring the data, assessing how are different types of what are the shortcomings of the data, more like those sort of analysis, like something that is unforeseen or nobody knows about this, exploring those areas. Whereas data analysts, typically they start from quite a good, clean data, but brings in a lot more analytical insights out of it, and making it like a presentable in a visualization and all this manner. Whereas a data engineer role starts playing so let's say some data came in the form of PDF, some data came from the form of text files, some are in CSV, some are in XLS, a lot of the different types of format, some might images, or some downloaded from the web. So how to clean this data, store them into databases, and make them easily available for data analysts and data scientists. So that's typically the role the data engineer plays. So they transform the data, clean the data, and then put it in a nice manner for downstream analysis by these 2 people. That's the majority of the expertise data engineers go. And data steward is the more the admin role. So what the admin role is, especially for healthcare, I can tell you, healthcare is probably the most regulated data of all kind of data types, because it's a very sensitive in nature. Although in India we are not yet that level of sincere, or that the regulation is not in that strict, but in Europe and US it is tremendously strict. So first thing is, all healthcare data, if shared between one party to the other party, that has to be anonymized. So there cannot be any particular name or specific identifier mentioned. So the data steward has to ensure that, let's say, if a hospital data is going to a insurance providers, because the insurance provider want to know how many insurance claim has happened from this hospital and those things. They have this legal framework. So the data steward has to ensure that all data are actually anonymized. Yeah, there will be definition about gender, age, and all this, but nobody can link it to our Aadhaar or the social security number or the name of it. So nobody knows whose data is this. It's totally anonymized, something like that. Then the second level of stewardness is to do an internal authorization. So let's say a data engineer might have access to all the raw data, but the data analyst and the data scientist won't have access to the raw data. They will only have the data which is in the database put forward by the data engineer. So the data steward has to make sure that these 2 guys does not have the access to the raw data. And everything, as you can know, will be stored somewhere in a soft copy, like a hard disk or somewhere, or in a cloud database that will be stored. So there has to be a proper data access and authorization in place that each people has access to whatever only they need, nothing else. So data steward actually plays those governance roles. It's very legally right role, but a very important role. And finally, whatever role you are playing, one of the most important aspect of being a data scientist is that data storytelling. Why it is important is that because it's ultimately your stakeholders who you are catering to. And why I'm saying is that, so we all know the McDonald's burger, right? Most of us like it. And whom we are catering to. So you might knowing that McDonald's actually customizes the burger based on the country they are selling or the taste they are selling. So even in Bengal, what you get will actually be very different from Bangalore where I am now. Or the taste of the burger will be totally different if you are buying in New Zealand versus US. So there has to be a lot of customization. So your end user, are you speak as a data scientist? Am I conveying this discussion with my fellow data scientists? Then we will probably be going in a very technical manner, and we will be discussing like, okay, we know what technical questions we'll be asking about. But if I'm speaking the outcomes of my data science analysis to like a decision maker, let's say a director of certain department, and that person might not be actually a data scientist. He is here to understand whether the drug they develop made some profit or not. If profit, how much level of profit that means. So they do not really need to know what analysis I did, what are the statistical test I did, and all these things. Those will be done. Those are for my fellow data scientists. But I need to provide him like a exact key insights that I got out of the data. So those are like a data storytelling. Similarly, like we in our life, we know if we have like we are telling a story to a 2-3-year kid versus we are telling some story or something else, or trying like a young or like a little somebody who's plus 4 or 5. We need to customize those stories. So the most important role and where most of the data scientists or any of the data geek guys fail is about data storytelling. But this is the most important part. If you can't sell what you have actually done, it is of no importance or no value to the organization. So that's the important part. And then how the data science daily life looks like. In movies or any of those things, it might look like, okay, these guys either they do day-night coding or something, or they do some line of code and they get some massive insight which like everybody clap about. Real life is a little different. So around 20% time actually gets on collecting the data. So you can say, okay, the data engineer has actually already collected the data and made it in a useful manner. Why it is so much painful, like I need to spend 20% of the time collecting the data? We will discuss in the next slide. Just keep in mind that around 20% time is spent in collecting the data. Second is cleaning and organizing the data. Why cleaning and organizing the data is that, okay, that's probably the role like a majority of the data engineers play, but the data scientists also need to play some role. And depending on the organization, if there is no data engineer, that the data scientists also need to play the role of a data engineer. Then we know that it actually comes up. And many of the times that's actually the reality. The roles are not so well defined. It's always overlaps a lot. So we spend a lot of time in actually doing data cleaning. So 80% actually time goes on collecting the data and cleaning the data. So why it is this kind of scenario? So let me tell you that. So let's say there are two insurance providers. I'm giving an example of a healthcare provider. We know that like a Star Health is an insurance provider in India, whereas HDFC Argo is another insurance provider in India. I'm just taking names. I'm not promoting any of them. So HDFC might have a unique system of using their unique identifier for each patient. So let's say I have insurance in HDFC. My unique identifier is XYZ 333456, but that refers to me. Whereas my company provides me insurance to Star. But Star Health Insurance will actually be having my ID as like a 345 Amit 0077. So those two IDs actually referring to the same person, but they have two different unique IDs. So you cannot merge my data using the unique identifier because the data is so unique identifiers are different and there is no unique format. Whereas my other parameters like my age and age and certain other things that they do not see, but they're like there is some common pattern. So you have to understand, okay, what are the common fields between these two different sets of data that I can use actually to merge the data when the unique identifier is different. So as you can understand, there are so many types of data and they will vary, so you actually have to spend in those kind of things. And then the fun stuff or the interesting stuff is actually totally 20% of the time. So mining the data, refining it, building the training sets and the algorithms and the analysis and the outputs and the visualization. So 80%, it's like cooking or something. So our preparation takes 3 hours, and then we finish it that in 15 minutes. As a data science role is also like that. 80% time in cleaning and collecting, 20% doing the so-called fun stuff. Yep. So now coming to the part. So like why I have linked it to, because I love actually cooking, and I often say like a data science is much like a cooking. So is that, so let's say you have a rice. That rice can itself be eaten as a rice, or it can go to prepare a biryani, or it can go to prepare a fried rice. All three has a different taste, flavor, and they take different amounts of time to get prepared. So the logic is, as a data scientist, let's go to case studies, and then we'll be actually discussing what the real life questions look like and how we solve the question. So the first thing I said, like we need to spend around 60% of the time searching for the data and the cleaning for the data. Let me give you one question or one real example of that. So there's a company who had a question like they had, I can't name the actual gene, that's like a business secret we can't reveal. So they have two genes that are known to work in MACE, major adverse cardiovascular events. And there is a gene X and a gene Y. And they wanted to understand whether gene X had a greater impact on gene Y, so that they could actually develop a new therapeutic drug against gene X, which would work better than gene Y targeting, because the targeting drugs on gene Y is already available on the market. So basically the logic is create a business scenario, or let us know if there is a business scenario at all exists, when in which case gene X actually has a greater potential to be effective in MACE. So if you search, I am showing you some data from the gene expression omnibus data, the GEO data sets, which you might have already used or like known through during your courses. So if you search for MACE or medial adverse cardiovascular events, you will get one or two data. Because MACE itself is not a term, it is actually a collection of disease. So like a coronary artery disease, atherosclerosis, acute MI, STEMI, so many collection of diseases that actually are actually related to MACE. So now you can imagine, even of the data of whom there is a lot of data engineers working in GEO who have made this portal available for the millions of researchers or the whole 7 billion people on earth to use it. So there is no way we can say that data engineers or the data are not properly arranged. But if you search for MACE, you will not get the results you need. So basically what you need to do, you have to go and individually search for each of the data, and now find how many number of studies are there, how many controls are there, how many tests are there. And as you can see in GEO, they use a different number of platforms. So which platforms are used, and each platform will actually have their own kinds of identifier. Some will have like a mentioned as a gene, so that will have the gene symbol. Some will be mentioned as a gene symbol. Both are smaller case and there is a space. There will be some like a gene underscore symbol, G uppercase, S uppercase, S, there is an underscore. So all possible varieties different companies will be using. But you need to actually collate the data for all these studies. First you have to search and then collate. So you have to write a custom line of code for all these and then fetch the data. So that's what I am saying. Even though apparently looks like the data is very organized and structured, yes, it's much better than manually collecting the data from each of the researchers. There's a portal that you can go and search, but there's a lot of steps that need to go downstream of it in order to make the data useful for further analysis. So that is why this 60 to 80%. So this 20% is about searching the data, and then the 60% is about collating the data, making them. There are some things you have to go read and study. This you can't actually automate. Whereas both have been done by like a microarray data, and those seems like a coronary artery disease they're talking about. Both are coronary disease, but they use a different systems to categorize the patient. One is the 3-condition experiment. So like in this particular case, we have to consider 0 versus 2. Whereas this is a 2-condition experiment. This is like there is only case and control, there is no normal patients. So we actually have to discard this. Those things you actually have to do manually and then filter, and then you have to go and understand, okay, should I consider 0 versus 1, 0 versus 2, or 1 versus 2? No, which patients are labeled as 0, they are the negative control. Patients who are controlled as 2, they are the positive control or the disease patients. So I need to actually consider patients labeled 0 versus 2. So this is where like a data cleaning and data arranging goes, and that's what the time looks like. Yep, let's go to second case study. So this is like a company which have a drug, like they have a target. They don't yet have a drug. They have like just like a target, basically, which they want to know how many number of disease this target actually can work on. So in pharma, there is something called like a in-licensing or out-licensing. So what is in-licensing is that, let's say I'm a small company, a boutique company. I have few people. I identified gene X can be targeted for disease Y. But I do not have the capacity to build a drug onto it, but I have validated it using preclinical rat or mouse or rabbit, this kind of preclinical studies. So what I can do is basically, I can sell this target with all the data to potential large pharma or the clients who has the capacity to build it. So I make money out of this. Yes, as business, you have to make money. And the other company, the big pharma company, without spending on the preclinical R&D, they get a target. So this is the company who is buying it. For them, it is called in-licensing. A company who is selling it, he is like out-licensing. So that's how it goes on. So let's say a company who has a target X, and they want to understand in which disease target X can actually work as a better, because there are some studies which say like this. So there are now actually more than 7,611 indications of the diseases recorded, including rare indications, and more than 311 human pathways known. Obviously, it's impossible for any companies to have, even if they have the best of high throughput screening availability, that they can scan 7,611 indications. They'll go bankrupt in less than 3 months. It's just impossible. So these are the questions the bioinformaticians or the biological or the pharma R&D data analysis to do, they get asked. So how do you solve it? I'm not going to much in the past details. I'm just saying like what we actually did and how we did it. So basically, there was like a 7,600 indications, and we know the target. Let's say a drug or that has certain targets, and the targets are like have some pathway they will be related. The genes are always working a pathway, and each pathway has an interlinking to another pathways, and that's how all pathways are linked to each other. So basically, using a systems biology pathway, you can shortlist certain indications. So we used a gene ontology and systems biology pathway-based overlap with some p-value, statistical significance to make it. And then we came out somewhere like a 200 plus indications. So as you can see, the immediate jump from 7,600 to a little over 200 indications that came out. But even testing 200 is impossible. Most of the pharma companies will test like a 10, 15 at mass, at that kind of range. Not maybe 15 is even too high. So can we even narrow down from 200 to like say 15, 20 such cases? So we actually built a machine learning based model using the clinical trial data, historical data. That's like the sales data or this online sales they use, our historical data, what we bought previously to predict what we can buy in future. Similarly, we can use historical drug performance data, drug disease performance data to actually potentially predict whether this drug is okay, at least theoretically is going to be successful or not. Definitely there are a lot of things that are unknown, but we can do. So we actually build a machine learning model. And then we also had to test it and then take the most successful. So finally, actually, and then obviously, we have to always provide the biological pathway related information. Like you have to create a rationale. I am saying drug X can work on cystic fibrosis, whereas why drug X cannot work on other fibrosis, like a pulmonary fibrosis or idiopathic pulmonary fibrosis. So those obviously we have to always support what we found using the biological information or the pathway relevance information, those kind of things. So in this approach actually gave like a 7,600 to 17 indications ultimately. So now you can imagine there is like a 99.78, almost 100% optimization of your search space. So if you will have like you have to find a needle in a haystack, and now you have to find a needle within haze, but it's like in a bowl of haze. So that's so easy. And it only takes around 8 weeks with 4 people. So that's the kind of optimization time, cost, resources can actually be saved using bioinformatics and your data science. So what we used in all this, simple same bioinformatics data, like I use like the R or Python that you use, those other tools have been used, and so many bioconductor packages also that you sometimes hear about during your courses. So those things actually been used here. Nothing exceptional. It's all something. It's just that, as I said, like with the Biryani experiences, we customized it for this particular case study. Third case study for us. So what happens is that when you market a drug, especially with cancer drugs, It works. It definitely works. But sometimes we see that the cancer, or like even with like the, or what we say, antibiotics, there is resistance that gets developed. So a company actually wanted to understand why resistance is building after some time, or is it what is happening. So then they shared with us their expression data sets of patients who develop resistance versus patients who actually got benefit out of their therapy. And analyzing this, we found that whatever happening in the body, that will have a subsequent analysis. We found there are some mechanisms that remain constant, but some unique ways of bypassing is actually gets developed within the resistant cells, which are not at all present in the sensitive target. So we to remain in being like a sensitive to the drug for the therapy, the pathway should not change, right? But we found out that at least based on the data, there are some unique ways the bypass was happening. Whether this bypassing is because of, we know in our body we have a number of isoforms which can compensate and those kind of things, that definitely they will test. But we gave them a number of different gene-gene interactions as a form of network and all, and we showed them how the network is changing around the drug target, so that they can do the next type of analysis. So this is how bioinformatics or the service industry provides insight using data or analyzing data. So these are all data science, but since we are using it in the biology field, we call it bioinformatics. That's what I said, like you use your biology domain doing data analysis or data science, that's bioinformatics. Now I will switch to some other ways where actually bioinformatics data, like data analysis is used, but it's beyond biology. So it's actually from biotech data. So the question was that there is a potential customer who actually wants to use some of like a new compound to better yield. So there's an oil producing company, and what they want to do, like if they use more enzymes, whether the oil production is going to increase or not. But as you know, like before you go on buying something, you want to try it. And that's when the selling company asks, so how should we actually be doing? These guys have sold us our 6 hours or 3 years of data. And what we found out is basically for that oil production company, these are the months, and this is how much oil they produce. So something they call the oil extraction rate, OER. We just used it. We see a huge monthly variation. It's like a periodic manner. So we see a lot of oils are coming during like January, February, March, but April, May, June, July, it drops. And the drop is very significant. Like if you see 6%, 16 versus peak around 2021. So like a 5% difference, or 5 by 16, like a 25 to 33% differences. Whereas the extra enzyme that is going to provide is around the range of 1%, 2%. So you can say, why would someone going to buy 1%, 2%? Well, 1%, 2% industry makes a lot of sense. It's like a for this particular customer, that 1% yield was equal to like a 5 million US dollar, around 35 crores of extra revenue per year. Or also that amount of oil worth around 35 crore, which was previously could not be expressed, but using that enzyme it can be expressed. So like a more benefit. And these obviously extra money translated to benefits not only employees as well as the farmers who provide the fruits for seeds for extracting the oil. So when we looked into the data and their trial plan, we did simple correlations analysis, simple Excel plots, some of the things, not much, and some R-based simulation. We actually found that there is some discrepancy in their data. That if you see here, some days they exactly show the same value, which is very like an oil potential. So oil potential is something can't actually be same. It's like and we found that there's like a up to 2 decimal points are same. So this is like a operational excellence can be increased. Whomsoever was collecting the data at the plant side, what he was probably doing, he was doing it once in 2 days, 3 days, or like once in 7 days, but copying the data for the same days. We are not going to blame that person, but these data are actually useless data. So we can only take one data out of those. So this is like we give them suggestions that you need to improve your operational excellence. Otherwise, you are losing a lot of insights. So that's something even data science can help you improve in your operational excellence. So let's say someday your oil is actually 20 point, potential is 20.15, but previous day it was 20 and you have all copied 20, you have actually underreported it. Similarly, you can inflate it for some days. So those always hide you real data. And with data science, the topic is garbage in, garbage out. So if you provide garbage to the analysis, the outcomes will be insignificant. So this is how we not only provided them the operational insights, we also found that there is certain change in trade because there are 2 mills that where they are going to do the productions. And if you can see the each line for a 1 mill, somewhere during the September month, or for whatever operations they're doing, there's a switch in the mill output. So till September, for a quite a good period of time, around May, June, July, August, one mill has a higher output, whereas the output of that mill drops quite a bit for the rest of the month. And during this September, there is a switch period. So we said like, okay, if you are doing a trial plan, make sure you do separate benchmarking trial for some period. So like up to September, you run one trial, and post September, you run a separate trial. So this is how you actually bring in insights. So like, so this is like a data cleaning, exploratory data analysis, but it helps you engaging in the customer, because the customer is not doing anything. They're not paying a single money for doing this analysis. It's that we will be selling the enzyme to them to make more, to like a more oil, and that's how we can make money. We are helping them in getting insights more out of their data. So that's like a more customer engagement. And that also, we are also helping both the customer and the seller to actually making decisions. So this is a different type of data that you don't see in pharma R&D, but this is also real life data analysis, and how as a data scientist you can actually bring in insights and help your both customers and sales. Second is, so this is actually a plant which is a based in a, like this is the same oil producing plant, small plant. It's based out of Malaysia, out of nowhere. So I have been there. It's from like a Kuala Lumpur, 250 kilometers from Kuala Lumpur, in the middle of nowhere. There is a plant all surrounded by palm plants, everything, beautiful area to look into. Why it is there? Because it was producing palm oil, and that's why what the palm oil is. So Malaysia and Indonesia produces 85% of the world's palm oil. So that's why it was in Malaysia. But the company's headquarters is actually based out of Denmark. Part of the decision making happens in US, and there is a potential customer in Brazil. So we have to link like the data that is being produced here to be live available to the decisions maker, and so that it can also be used for sales purposes. So we actually had to build a data ingestions and on the fly data analysis, and ultimately creating a visualization. Like small, they do not really need much insight. They really need like a one or two. But who is the mill operator, for his efficiency, he needs like a daily fluctuations data, daily variations data. So you need to take into account who is your stakeholder, which slide will be used by which person. And all of this has to be on the fly. So each day new trial data goes on, the data gets updated, and new insights come in, and it says like how it has deviated from the previously, whether a trial that happens for 15 days, how much the outputs are like this. So the mill operator will look in every day, but the decisions maker or the buyers will look at end of the trial like once in 15 days. So they need the summary output, whereas the mill operator needs a daily detailed output. And there's like they all had like an Internet of Things IoT sensors there, and around 1,300 different types of data are being used, ingested every 5 seconds. So this is how you make life easier for people. You build for certain pipelines, you make things robust, and that connects the whole world together and helps you making decision making. So that's how business moves forward. This is the value of it, brings it to the business. It just accelerates everything. One more case study is that it's about measuring like this is, I actually worked in palm oil for around 9 months or so. So some of the case studies I have got from here. And this is what happens in the lab. This is not actually in the field or in the customer or in the trial plant. So these are cells, different oil bearing cells, and these are different oils. So basically if you overlap them, some of this point will actually be inside this. So this is how each cell contains some oil, whereas these green dots are like the oils that is outside. Those can be collected within the vial but not within the cells. And these blue dots are like a junk. So if you, any of you might have already have like some idea of how fluorescence microscopy works. So this is like the cell debris, the small dots are big on around the cells, whereas the other channel, the green, which is a fixie stain, it was like a oil. So we wanted to understand, like after some process, how much oil is actually stuck inside the cells, which could not be extracted, versus how much could actually have been extracted. And then we can see using the enzyme whether we are extracting extra oil or not. Because enzymes are known to, there are certain enzymes which will break the cell barrier. So ideally the enzyme should actually break the cell barriers. And what happened is that those guys used to take these images, then they used to use ImageJ, a free software, or many of you know it as a Fizzy. So then they do. So this has been a manual process each time. And it was sensitive to who is doing. So if a person who has been doing it for 3 years, the efficiency or the accuracy of the person will be much higher compared to an intern or like a new hired guy who has been there like less than 3 months. So the consistency was also fluctuating. So the idea was to automate image data analysis to increase the throughput, because if you are doing it automate, so a human was probably doing 20 of such capturing images and analyzing, but if you are automating it, we could only the capture has to be done, the person, even that could be automated, but we scaled up from 20 to like 350 or something like that. So two people are capturing 350 images, and even before they capture the second image, actually the analysis used to happen. So how you do is that it's a backend, so you convert the image to a grayscale, then you change the contrast level, and you figure out, okay, give me the surface area and take out the larger surface area so that you can remove the debris. So this is what you are doing still in these cell images, like this particular image, not this. So from this, you extract the cells. And this cell boundary, so boundary is all about, it's like XY images, we know the pixel numbers, and these pixel numbers actually overlap on these particular images. So this particular boundary now gets overlaid into this boundary, and once it is overlaid, we can now say, okay, give me those black spot that is inside the white spot, like within the cell boundaries. So from here, we are now able to extract the oil that which is within the cell level. Now everything else which is not within the boundary but in this green channel is actually an outside oil. So now we can mark it, okay, the red is actually the cell, the green is the oil inside the cell, and the blue is the oil outside the cell. And that is so fast, like it hardly takes, I'd say, 2 seconds. And these images are big, these are like a 16 megapixel, 28-bit images. So these are quite big, but it takes less than 2 seconds for a simple laptop to analyze it. And whole this office has been actually coded in Python. So this is how you use a data science, simple coding logic, and some of your logic that you as a biologist, because you have to be a biologist, or you have to know and understand how fluorescence and the fluorescence microscopy works, then only you can use it. So use your biological knowledge to build a solution based on data science. And it's simple, just coding. There is no magic thing happen. So now I think this is last case study that I want to talk about is storage of information. So one of the things is like I have simply taken like a PubMed information. So we have PubMed available, right, at our end. Why do we need to store PubMed? No, we sometimes need to store PubMed. It's several gigabytes, the download, even simple the abstract. There are thousands, like even hundreds or so abstracts, hundreds of thousands of abstracts. Now the point is, let's say I wanted to search for thermostable and let's say oil palm, something, give me all the articles which is written as oil palm, and if there's something like a thermostable or cellulose, something is written into it. That's how we write in PubMed, right? We write oil palm plus thermostable. So sometimes what happens is that in organizations, data access is like there's data stewards, the governance at a different level, and it actually slows down the process. So what organizations do, they intelligently extract the important information out of the data and store it more efficiently. So what it does, let's say we feed the abstract of each article once it gets published. So let's say every week, Friday, when everybody is in the computer and automated script runs, it fetches the abstract automatically. And then from there, it fetches the strain name, then certain text or the parameters that are of our interest, and tags it to that particular abstract ID and store it in this format. So let's say rather than storing this whole content, it is saying that PMID, organism name, organism type. So it found it's a bacteria and it's a map. So it is not written anywhere that bacteria, but it has figured it out. Is oil palm written? Yes. Is EFD written? Yes. Is the terms, what are the tags, thermostable and endonuclear. So natural language processing or this kind of things can actually be used to extract these tags. We call it tags or the annotations, and then store this particular information. If I need the author, okay, I get the ID and then I can do. But I do not always need the authors, right? I always need only the articles, the content. And what's the benefit is that if we are going to store this whole information, it takes 1.16 KB. And now we are going to say, these days we talk about 256 GB phones, 1 TB phones. Why do I care about 1.16 KB? The reason is you have like let's say you have to search among 2.5 millions of abstracts and each taking 1.16 KB. Then you know the scales you are at it. So if it takes less than 10% of the space, nobody cares about the space, but the speed increase in search output is so fast. So it optimizes the storage, is a very fast query, it is scalable, and those it can be easily integrated to the downstream application. So each time nobody needs to read it manually and feed this article. We only can extract information from this kind of information, and then use it for our downstream purposes. So like this is how text mining or the pipeline automation and this works. So you can write a script that every Friday evening do this when nobody is at office. Computers are free, resources are available, but by it gets ready by Friday evening, but on Monday morning when everybody comes home, they need the data, it is available to them. So that's how actually data science is helping transition different kinds of industry. We discussed about some of the industries like weather or the healthcare. I have given you some examples of like biotech or like this text analytics or like how pharma works. So these are some of the examples of how data science is helping in industry. The idea was to give you some insights and enthusiasm, create some enthusiasm that, okay, these are the things that I can do as well. So the next question is how to become a data scientist, the million or the billion dollar questions. If today I want to be, how should I become? I guess this is actually today morning I got in my LinkedIn feed. Okay, then I downloaded it. And this is what I searched in Google, how to become a data scientist. Okay, somebody is saying me they can train me in 50 hours to be a data scientist, just like a little over 2 days full, but I can't work 2 full days. So even if I spend like 5 hours a day, in less than 2 weeks, I will be a data scientist. Okay. They are saying that they can make data scientists, which will give them like a 50% average salary hike. So if you're earning today 3 lakhs, tomorrow you'll be earning like a 4.5 lakhs or something, but it's a 11 months course. Next, someone is saying they can make you data scientist in 6 months. Next one is saying even less than 3 months, and this is high. This can make you 50 hours. So if you're going through all this, you win this is actually not how to become a data scientist. This is not how it happens. Nothing happens in a day. I have a 20% body fat, and somebody giving me a slim, lean body in 1 week. It just doesn't happen. It's like all fake or fake. It's mostly marketing to catch the attention, and then when you sign up, you will get the reality. But to become a data scientist, it's nothing like a rocket science. Even rocket science also uses the same things these days. So it's a very simple but a continuous, I'll say, a dedicated process. By 3 months, you will definitely get some idea. By 6 months, you can say, okay, I have the basic fundamental skills. So it's like 6 months, it takes the time to make a transition from any field if you have some basic idea about programming. That's the starting point. It could be C, it could be C++, it doesn't matter. You need to know what is a variable is called, what is a loop, for loop, while loop, if loop, or the logic statements to be written. Any language, you just need to feel like those logic flows into mind. That's if it is a static point, then it takes around 3 months to 5 months to just get rolling. And by the time you can call yourself, if you're dedicated, a basic level. So what are the basic skills required? These days, the primary analytics skills are like R and Python based. So Python is still quite popular, but there are other things like a SAS, but those are not so well. So if you know R and Python, you could be saying, okay, I have the basic programming skills related for data science. R Python itself has a lot of visualization packages, but investigate something. I have used Tableau as one, but there is also Spotfire. There is Microsoft Power BI. Any of those visualization tool can actually, you need to have a basic idea. So Tableau gives like a 15 days of free license or something. You can download it and you can use it. And then Tableau Web is always free. R Python is totally free. SQL, you know SQL, basically some database of knowledge. You need to have some database related knowledge so that you can at least extract the data or the store the data. Cloud these days has become a little, so there is like a Google Cloud, GCP, Amazon Web Services, AWS, and then there is what is the other, Microsoft Azure. So these are the 3 popular cloud platforms. You do not really need to be cloud expert or do not to pay anything. There are lots of free courses or free YouTube videos and knowledge. You need to know what is cloud. It's a cloud that is on top of our head or something like that, or it's what it is. How high people talk about cloud and all this, something, and some basic fundamentals of machine learning. There is if you search for a 50 popular questions in machine learning, you will get a lot of PDFs. So those kind of things. What are the machine learning algorithms? What are the requirements, types, supervised learning, unsupervised learning? What are the types of supervised learning, semi-supervised learning, iterative machine learning? So those basic. So if you know, have a hands-on between R, Python, and Tableau, basic level knowledge of SQL and cloud and some fundamental knowledge of machine learning, you can have there. I have the basic skills of data scientist. Yeah, basic skills are also these days in demand. So basic skill even can give you a job. That we can say. Then how do you become advanced? So when you have the basic skills, then you start to get into next level of things like the natural language processing. What I said, if I say, give me all the patients who are diabetic and give me all those adult patients who are diabetic and using statins. So can the program make a query from this? Okay, by adult it means all the patients age either 18 or 21 based on the country. Diabetic means it will include both type 1 and type 2 diabetes. So it is talking about the two disease name. And statins. So statin is a type of drug, but it has a lot of brand name and generic name. So extract these particular terms from the sentence and then make a query or something. So the extraction of the terms or something or the relationship on those are done through natural language processing. So if you're saying you're advanced, you cannot be advanced on everything, but let's say, okay, you know advanced about natural language processing, or somebody can say GPU computing. So we use GPU for gaming. Many of you might actually know, and we're famous for about we have fought hard with our parents to buy us like a GPU or something because the frame rates doesn't work or it's slow or the graphics is not that good without a GPU. But GPU has a completely different purpose in data science itself. So GPU are actually uses as parallel processing computer. So if you have a AMD or like i3, i5 or i7, so how much core you can have? If you don't go to Xeon processor or something, at max you can have like a 12 cores or something, and with hyper threading you might have 24 core to process your data. But GPU, they are like a lower speed cores, but even the basic GPU these days will probably also have 128 parallel processor or something. So if you see, it has a number of more processing power than a CPU, but it cannot do high level tasks. So small level parallelization tasks can actually be done using GPU. Simplest example is like if you have used any of your courses RASMOL or PyMol to render atom or like a protein structure. If the RASMOL or PyMol when installing, if they detect there is a GPU in the system, it renders much faster because then simply the rendering each atom is the same level of code. But let's say you have like a 1500 atom, 1500 atoms to render or create the 3D view. So the CPU it's like a 10 core CPU. So it has to process each CPU has to process 150 times. But if you have, let's say, around 150 core of NVIDIA GPU, so along with the CPU, 10 plus 150, 160, it has to process only 90 times or so. So that is how the massive speed increase actually happens from GPU. So similarly, let's say if we go here, we have to process text data, and I have 1000 text to process. My 10 core CPU can do, but this 100 core GPU can process each text parallelly. So that's how the speed increase happens in GPU. So GPU computing these days is taking a lot of news, and there's a lot of things, especially image analysis happens a lot through GPU. Many of the software robots or the physical robots that you see in sorting systems or like the car manufacturing, they actually use GPU to process the image that being captured by the robots. And then there is this deep learning libraries or the linear algebra, basically the linear algebra libraries that are used in deep learning, PyTorch, TensorFlow. There are many others. So those are they will ask you if you say advanced, they will ask whether you have a advanced level skills in what are your advanced level skills. So acquiring skill sets is more like you are going through a syllabus or a course, but where do you get the real life, near real life experiences? That's one when we give how good our knowledges are. That's what our exams are about, right? But here we do not always need to be evaluated or something. But if we aim to get a real life experience, where this data is available, because data is not always free. So there are some competitions goes on like Kaggle or Machine Hack. No one has to win those competitions, but being participant here, you get access to the data and you get to see how others have solved it. So that's how you get a real life experiences. Like few days back, there was a protein data was available in Kaggle. So there's 67,000 of different cell images being captured, or 4 or 5 different channels, and they wanted to use data science and image analytics to classify those protein to those cells based on the proteins that they are expressing. So this, you do not have real access to the data, but if you are a participant in the Kaggle, you have an ethical access to it, and you use it for your development or like some. So, and you also get test your coding skills as well, and you can say that I have participated in Kaggle. This is how you sharpen your skills. So this can give you near real life experiences. Why I'm saying near real life experiences? The reason is that most of the time these are very well cleaned, and the aim is to improve the accuracy of analysis. But as I said, like in a real life data scientist, 60 to 80% time goes on cleaning or preparing the data. That's what you do not get. But the 20%, which is like the epitome or like the cherry on top of a cake, those experiences you can get from Kaggle and Machine Hack. And if you really need to get full real life experience, the best is to get an internship. Be paid, it could be unpaid, but definitely you do not have to pay. These days there's a lot of internship. Everybody is willing to hire a lot of interns who want to do, like it can be 2 months, 3 months, or 6 months. But this is where you actually get the real challenge. You have to do everything like data cleaning, preparation, storage, everything. It could be one small experience, but this is the most valuable experiences. So this is where I would say like the path, you can say, or the stages to create yourself a data scientist out of something. And obviously to get ready for industry, I think this is the slide I also talked last time. There's some soft skills are always required, and this you have to develop in parallel. Your communication skills, like how you communicate your timelines, data analysis plan. It could be scientific, and as like whom you are talking to. You could be talking to a technical person, you could be talking to a stakeholder. Definitely there needs to be some motivations, some ownership or the leadership capability. Let's say somebody has given you a data on Monday and asked you to do something on get the output on Friday. But Friday when he asks about, you say like you did not get anything out of it. But you might have genuinely tried, but you did not get anything. That's possible. But why did not you communicate in time beforely so that the person had a like altered or like a different timeline of expectation or could have helped you out that. So this also builds like a taking responsibility to deliver time or proper communication to create a teamwork scenario. But obviously you need to solve some of you. There will be assistance in industry definitely, but you need to solve some of your problems. Every time you are stuck and every time you are asking somebody to help you out, then they will ask you your credibility or competencies. So a problem solving attitude, a decisiveness, and sometimes once in a while there's like a tight time management. That's always happens. Even as a student, some chapters we are unable to revise or finish even before the exam, but we do somehow manage. So like the ability to deliver under pressure and time management. Flexibility as a data scientist is actually most important because as I have said, it's like you need to have your skill sets, but you need to be flexible based on the audiences, the data, the analysis, the outcome. You can do a very deep learning analysis, very intensive analysis, but it does not solve the business purpose. There is of no value. So being flexible to it, to the business outcome and the goal is the most important. And obviously the ability to, this is a real real life capability. It's like a negotiation of the conflict resolution or something. Not always you will agree on something, not always others will agree on something. So how do you find a mutual ground? These are some of the things that will definitely help you to build the secrets of successful career progression or the career studying. So these are definitely very required in everything, whether it's industry or any academic career as well. Yep. So that's pretty much from my end. Thanks for listening. And now it's open for question and answer.
Yes, thank you, Dr. Das, for that great session. And it was indeed a very informative session. I hope students will be highly benefited from that. So if you have any questions or anything you want to ask to the expert, you can unmute yourself and ask maybe one by one, or you can put it in the chat box also.
Sure.
Yes.
I'm Ashkar.
Yeah,
So,
sure.
Yes.
explain me for biotechnologist
Mhm.
or particular bioinformatician. So, in data scientist, data science course and what are all the essentials that we need to learn?
Yeah.
We want a profile to upgrade in bioinformatics platform.
Yeah. So basically, I would say first get those skill that I said, like get a quite good coding skill, hold of R and Python. Those two are like the bread and butter these days. And parallelly, get some experience with Tableau, Spotfire, or any of the visualizations tools, because you need to communicate whatever data you are analyzing in the form of a visualization or the slide set or something. If you are what are your endpoint results? So R, Python, Tableau, and by courses, I would suggest there are three, four things that goes on these days, like the upgrade and many other things that you simply learn. You get a lot of advertisement because they charge is very high, like a 50,000, 1 lakh or something. I have personally paid 0 rupees to upgrade myself, because in my whatever is part of it was definitely on the job learning, but I have seen like there is a lot of YouTube videos which is sufficient enough, or to get you have to form the query. Okay, R basic tutorial, and you follow that tutorial. That will give you some headline. Or you can say R visualization, data visualization tutorial. You will get a lot of web resources. In those paid courses, they teach you exact same thing. And those are all online courses, not that some expert will come and will teach you. They will probably be, and they actually contact us sometime to be the expert. So you will get like a 1 hour sessions on a weekends or something. And for that, they will charge you 50,000 for a 6 months course or something. So I would personally say, if you have money to spend, go and spend. Otherwise, just use YouTube or free web search links and get some skill about R, Python, Tableau, and familiarize with yourself one of the cloud technologies. It could be GCP, it could be Azure, or it could be AWS, one of the cloud technologies, and some SQL and NoSQL. That's all you would need to start or transform yourself from a bioinformatician to the data scientist.
Okay, so there is one question in the chat box from Piyush.
Yeah.
Yes, Amit, can you take this up?
Yes. Yeah, thanks, Piyush. Do you think having a PhD is essential even for industry sector, reaching higher company roles, having some experience is being looked by the company? Yes. PhD, I would not say is essential to enter company. No, it's not essential. Even companies are open to like a BTech, Biotech, BSM, BTech, Biotech or MSc Biotech. Those are fine. But what happens is that it's with the career progression at some point. So let's say somebody with a PhD will actually start 1 or 2 positions higher than somebody who is not with a PhD. That's like the basic how industry has worked in general. But what also part of the industry, starting is one, progressing is another thing. So if you don't have a PhD but you are highly efficient, you will progress equally well. So PhD is not essential, but PhD, I would say, trains you on certain things. Now, what are the PhDs like? You do not really need to publish 10, 15, 20 papers. That's no even industry. It may not be related to the industry that you are applying. What would say you should, if you are doing a PhD, try to like how to formulate a question, how to what experiments or the tests to do, or like in this case for bioinformatics, how to analyze the data, get the insight, validate the data, and then present it in a scientific manner. So while you are doing a PhD, these trainings mostly goes on. So that's the reason actually a person with a PhD joins an industry 1 or 2 posts higher, is that this is a training that they have already gone to. The time or the investment required, not by money, but by the time or the mentorship investment required to create a person with that level of expertise is not required for the company. So that's the advantage you bring into the company, if you have, obviously along with your analytical skills and capabilities as a PhD, but it's not essential.
Okay, any more questions? If you have any question, you can unmute yourself and ask directly.
Yeah.
Okay, if not, then sir, George sir?
Yes, madam. Yeah.
So should we end this session or?
So we will go for 5 minutes break, like 4 o'clock.
Okay, so maybe you can thank
Okay.
Dr. Das
If
for
we are not
this.
disconnecting the meeting,
Okay.
no need to disconnect the meeting. Yeah.
I think I'll stay. That's a good deal.
Okay.
Fine.
To listen to,
Yeah.
and I think
I think,
one more question popped up.
Yeah.
Sharup, I found difficulty during microanalysis in model generation using lemma. Can you please explain lemma? Okay, this is quite a technical question. Explaining lemma. Maybe Dr. John, you can share me something. We can discuss. We can take this offline because this is quite a bit of technical. We did.
Yeah. Okay. Fine. Yeah. Yeah.
Sorry.
Yes, sir. We'll reply. Yeah. Yeah. We'll reply in offline later. Yeah. Some more questions.
What are the main opportunities after MSc Bioinformatics? Which companies hire bioinformatics students? Yes, especially. Okay. I had shared a previous slide. If we have time, can I share once
Yeah,
more?
sure.
Yeah. Yes.
So maybe you guys can take a screenshot of that. There's a lot of companies in India, they hire bioinformaticians. Let me just bring in my slide and share with you. Please, if you can, I hope you can see my screen. Please feel free to take a screenshot of this or a photo of this. Yeah, during our previous talk, we shared this. So on the left hand, these are all biology-related companies which have Indian operations in India. Some are India, some are international. I think these are all Indian. Pyrian is a US company. These are all major pharma who hire bioinformaticians in India. I know people who definitely work there. So all of them, at least all these companies, I know at least someone who works. These are IT companies. Christian Hansen is a biotech company and Novozymes is a biotech company. Unilever is to a lot of FMCG and many other things. But others like a PwC, Deloitte, these are management consulting companies. Indigene specifically caters to life science domain, but they all hire bioinformaticians, and these are all Indian IT companies, and they hire bioinformaticians for different. They do not have mass scale input like IT, but they do require bioinformaticians. I hope we can. You all of you have been able to take a screenshot, so I'm stopping sharing. Yes. Anything else?
Yes. Any more questions?
Hi, sir.
Yeah.
Sir, informatic.
Sorry, Ashraf, I can't hear you. Can you repeat and can you please put the mic?
Yeah, sir. Mic. Yeah. Lots of noise is coming.
Sir, bioinformatics application in R&D, like in drug designing. Can you just explain?
Yes, so that's, I think you read a lot about your curriculum. That's the reason I little avoided it, but it's still a major part of our bioinformatics being used. So that's like a structural design, structure-based analysis, CADD, these are used. Unfortunately, in India right now, no major pharma company has a R&D. There are few boutique companies. I know like Cellworks, Bagworks, which are based out of Bangalore, like a little older startups, 10 years old or something. They use, but they have small footprint here. That's the reason I little defocused it, because the standard drug designing industry or the R&D in India is not, unfortunately, that big. All big companies have offices here, but for that data analysis that they generate outside India. That's a little sad part. There used to be an AstraZeneca R&D center where I myself was an intern like in 2008, but that was also shut down in I think 2011 or 12, somewhere in that time in Bangalore.
One more question was there in the chat box.
What do you think of bioinformatics in agriculture? Since after completing agriculture, I took bioinformatics. Yes. Have you heard about Monsanto or Bayer Crop Sciences? They are the largest in globally, and they have the I would say Asia Pacific largest data analysis and the bioinformatics center, which they cater to all Asia Pacific countries from India. So there are competitors. I do not immediately remember the name. Like Bayer Crop Sciences is one of the one who hires bioinformaticians, or like people with bioinformatics knowledge from agriculture background is of huge demand there. So do look for in LinkedIn like the data analyst Bayer Crop Sciences. You will get people who work there.
Okay, I think no other question. Yes.
Yeah.
So how bioinformatics help in plant physiology or biochemistry?
Yes. So bioinformatics that I say is like a let's say even if this crop sciences example. So all crops they do breed for different kinds of scenarios. In some cases, like I have been to one of the Bayer Crop Sciences offices for one of their public days. So they let's say some countries require cauliflowers to be quite white, whereas some countries require tomatoes to be longer because that's what they sell there. But in African countries, they need to be the outer skin need to be in such that they can bear the heat and the long transport in the African countries. So these are some of the physiological parameters or the cellular parameters that needs to be exhibited by the particular crop or the plant. But when you generate the data, what you want to do, okay, what the genetics had gone into, why this plant exhibited this, what are the characteristics, and which one should I modify versus which one should not I modify, so that I can breed a faster or a better generation in the next generation, and that will help me to make business decision faster. So you gather the data about plant physiology and the other parameters, what watering system, temperature, water, and everything, or like the images of the plants, and then you analyze using your bioinformatics knowledge along with and pair it with their genetics to figure out, okay, these are the mutations or the modifications or the haplotype that has worked, that has not worked. So it's simply like use your biological knowledge, but the data is from the plant compared to the pharma data or something. It is there. I hope I have answered your questions partly.
Can I ask one more question?
Yeah, go ahead if you have time.
Microbes are pathogenic and non-pathogenic, both are available in our environment. And how bioinformatics help to identify which microbes is pathogenic and which is non-pathogenic?
Okay, yeah, sure. So I think not only in plants, in microbes research is actually big. Even I think you have probiotics, you have heard. So probiotic science is a big science going on. And but with respect to your questions, so there are certain genetic traits. So first of all, you can add, like say from a data analysis point of view, I'm trying to say. So you see your plants has gone through certain changes, and you see that there's a disease. Okay, you collect first of your images of the plants, then you isolate whether it's a virus or a bacteria. You isolate from those affected areas, get them sequenced or get them what are the properties there. So now you have the virus sequences, and you can do phylogenetic analysis to link it to the viral progeny, so you know from where it came from and what are the mutations that has caused this level of devastation into your plants. And you have your historical data or something that you can say, okay, these are the mutations in a virus or in a bacteria that is making it, like I say, even in a high temperature where other viruses previously the bacteria did not survive, but even in higher temperature, let's say in Africa somewhere, this virus is giving us the devastation. So this is how you are relating bacterial or viral genomics data with your disease data and the physiology and the biology to get insights what are the mutations are causing. So next time, let's say you have this data stored, next time somewhere in some other countries, so let's say in Middle East, they say like, we are reporting, or somewhere in India from Nagpur, we are reporting similar symptoms. So you immediately know, get it sequenced fast, and this is the trend and the pattern. If you are following this, then this is the virus and this is the treatment. If that is the pattern that is following, then it is the treatment. Because many viruses might have like a similar starting point, but they end up differently. Whereas the others could also be happen, different type of origin, but they end up similarly. So this is what only your historical data and the proper analysis and pairing it up with the viral and the bacterial genetics can give you a head start, or can actually help the farmers. Because if the crops die, then nobody makes a profit, nobody makes a living. So that's how actually the data analysis is helping these days.
Thank you, sir.
Yeah.
Sir, one more question can I ask?
Yeah, so go ahead.
Sir, this question is related to our previous Aga's question. For biochemist, bioinformatics work is one of the preliminary contribution, where biochemist can, especially in particular like ligand finding for inhibition work. So, economical viable as well as it is fast result giving, so that they can synthesize that compound and they can check the IC value. Sadly, I am from Karnataka, Hassan, near to Bangalore.
Yeah, I know.
Sadly, in Karnataka, like more of the biochemists are not focusing on this area, and bioinformaticians also are not approaching them, so that their research can get escalated and economically they can get a good results and they can publish a good.
Okay. Paper publish is something different. So because paper publish is whether that is scalable to an industry level or whether whether the IC50 value that you have shown, whether really that makes a business case or like a economical benefit, that we not go. But what you are asking is like a academic industry collaboration or like a cross expertise collaboration. So this is something that do not really in hand of the individual people. It is where the policymakers, the government, the academics. I think since you are here in Bangalore or the Hassan, you know like GKVK. And GKVK, that university has a quite a good collaboration with NCBS, and so some things in patches it will happening, and there is also ICAR. There has a lot of collaborations with them in the agricultural field. It will take time to happen. I cannot tell you that it is going to happen immediately or it's something. It requires an ecosystem. It requires a support culture to actually get the total benefit. There are things happening, but it will take time. There is no immediate solution, unfortunately, for this.
Okay, thank you. There
Yeah,
is
thank you, Amit.
quickly, we'll take few more questions. There is one question in the chat.
Yeah, quickly, already it is there.
Yes.
Yeah, it is there in the chat box. Quickly we can.
Yeah. So, good. What you think of bioinformatics in agriculture since completing agriculture? I think we discussed that.
Yeah.
It is hard to jump from MSc in botany with a planting taxonomy major and PhD in bioinformatics. I'm not going to say it's easy, but it's doable. I did zoology, masters in zoology, then molecular biology and bioinformatics, then bioinformatics, and then data science. It's doable. And as I said, like it's not going to happen 3 months or something. I would say like you make a viable plan to make the transition in a 3-year period or something like that. And that's what, since you're already into PhD, I guess you will get the durations to make the transition. So make the best of use of your PhD duration. Data analysis and data engineering, which area has more opportunities? They are interlinked. So I would not say it's something that is, and it's dependent on what kind of industry you are. I would say both has equal analysis. And as a data scientist, we need to know both. But if you are an analyst, you need to also know engineering. If you are an engineer, you also need to know who is going to analyze it. Then only you can make the sense of how the data to be stored. So both has equal opportunities. There is no shortage. And honestly, in India, 90% jobs will be like a data scientist. Probably 10% will have a demarcation as a data analyst or a data engineer. So it's not that any of them is lower or something. It's very essential to be knowing both. What is the main role of a bioinformatician in present-day biological research and development area? I would say the role of a bioinformatician role is like a magician or like a truth teller. You have, as a bioinformatician, you have the potential to accelerate discovery. So imagine what is the potential. Even I have seen like a high throughput screening in AstraZeneca. They can scan how much amount. If there is only one single protein, they can scan around 50,000 compound in 2 weeks or something. With some repetitions and something, and come to like a more short analysis. But with that bioinformatician to level of some sort of doing that analysis, or like even if you simply do high throughput screening with a ligand and all, and you do molecular simulation, companies don't, mostly large companies have a lot of computational power and something, they can accelerate to like 1,000-fold or something. And with this cloud computation, on demand scale up. So like when the demand is there, they can scale up to thousands of times, millions of times. When the demand is there, they will just shut down all the systems that they don't pay for it. So main role is to reduce the time, accelerate or optimize the output time, and then aid in decision making. So let's say if a company had like take 1 month to make a decision whether which other 10 cell lines a compound is working, using bioinformatics or the other data, you can probably do it in 2 weeks or something. Yeah, single, or if there is a more person, you can even reduce to another shortest time. So that's the person. And you can tell the truth, whether it is going to work or it is not going to work. If yes, why supported with the data? If not, why supported not with the data? Yeah.
Okay.
Yeah, it's good to see so many questions. Thanks all for asking questions. I like that.
Yes, we had that night session. Sir, should we conclude this session and maybe we can...
Yes, continue. Dr. Saram is there already.
Okay.
He joined. Okay.
So, yeah.
We can continue with.
Okay. So, Dr. Das, thank you. Thank you for that informative session. And I think students got really charged up and they asked a lot of questions. That was a very good sign. And so, we thank you for joining us and giving us your valuable time on this Sunday afternoon. So, thank you, Dr. Das. Okay. So, we'll move to our next session. Our next session, Dr. Sairam has already joined. So I will like to give a small introduction about Dr. Sairam, and then has he joined, sir?
Yeah, he joined just now. He left.
Okay, okay,
Let me
okay.
see.
Okay, maybe some
Maybe
problem. Yeah.
network issues. Yeah, he joined back. Yeah, he joined back.
Yes, sir. So I'll give a small introduction of Dr. Sairam. Dr. Sairam has his master's in medicinal chemistry from Rajiv Gandhi University of Health Sciences, Bangalore. He had a PhD in computational chemistry from Jamia Hamdard, Delhi. He has 16 years of experience in pharma industry, working in Accelera Knowledge Solutions, Hyderabad, Zydus Research Center, Ahmedabad, Sun Pharma Advanced Research Company Limited, Vadodara, Gujarat, in computational chemistry, supporting medicinal chemistry. He has expertise in API, biotechnology, and formulation sciences. Currently, he is leading bioinformatics and computational discovery at Sun Pharma Advanced Research Company. So, Dr. Sairam is here, and I would request her to take over the session. Sir?
Yeah, thank you, Subarna. Thanks, Dr. John, for this opportunity, and thanks everybody from the university for inviting me. So I think it's I have been hearing Amit's discussion. It has been quite interesting. So many questions. So what I'm going to do for the next 45 minutes or so is that I'm going to project before you some of the bird eye view that I have tried to compile with respect to different aspects of pharmaceutical industry. Firstly, I will not be able to focus only on bioinformatics. I think the applications have been limited to the extent that from India we have been like not extremely positioned to do core bioinformatics work that we often get to see from the international publications or the work that they generally publish. But yes, there are many companies who are using that, and we, to our limited extent, wherever it is, important, we keep applying bioinformatics solutions. And since the department that I am currently heading is itself is called Bioinformatics and Computational Discovery, so we have an amalgamation of both bioinformatics as well as computational discovery, where computer-aided drug design and techniques associated with that both are given. So now I'm going to walk you through some of these slides which I have tried to compile and show you. And then largely once this discussion is over, largely I want to keep it a proactive session, or rather discussion should be happening. So that's what I'm trying to do now. Okay. So I think one of the reason why computational techniques come into picture is just because of iteration rates in drug discovery. And I know, as somebody said in one of the conferences, that it is easier to send probe to Mars, but then it is difficult to discover drug. So because every human being is different, and the way each person responds to a given drug is going to be different. I think it's all the reason why it is more difficult to discover drugs which is suitable for all kinds of population. So the major reason why a lot of failures that happen in drug discovery are because of pharmacokinetics and bioavailability and efficacy. Efficacy is the effect for which the drug is actually made, and pharmacokinetics is how the body metabolizes the drug, and how much after pharmacokinetics, or how much metabolism that body has metabolized the drug, how much actual original drug, the parent drug, is actually available for the activity. So as you can see from here, the majority of failures are because of two major reasons. With any phase of clinical trials, one is major is efficacy, the other one is pharmacokinetics and bioavailability, and to some extent toxicology, that is toxicity of the molecules. So I'm going to take you through some of the key things that are encompassing different aspects of computers in pharmaceutical industry. As you can see, computer drug design. We will briefly discuss what is about this computer drug design, different aspects of it. And computational pharmaceutics is it is a formulation sciences, like there are tablets, there are capsules, there are syrups, and then injectables, so and so forth. So what exactly are different aspects of computational sciences which gets into these different formulations is generally or traditionally it's called as computational pharmaceutics. In silico toxicity, that is for developmental toxicity in terms of genotoxicity or mutagenicity and so on and so forth, are actually captured in this in silico toxicity. Informatics, CIML, I think Amit has taken you through different aspects of AIML and data sciences. And informatics to an extent like electronic lab notebook and all that, and then other chemoinformatics, all those things will come where actually you do fingerprinting and you do metrics to understand what is the similarity and all that. And finally, bioinformatics, where you use biological databases to derive sense of that biological information. So, now key applications, especially in basic drug discovery, that is where you are given a target or you don't have a target. Now, typically, these are the two different scenarios that it is. We will in a moment see what exactly different things that happen when target is present and what when target is not present. But the let me take you through the different aspects within computational drug discovery. One is molecular docking, where actually you try to identify the protein of your interest and try to fit in the molecules that you think are important into the substrate binding site or active site, slash active site. Any of you can call it by any times. But a substrate binding site may or may not be active site. So and there are allosteric sites. So there are multiple pockets within the protein where the molecule can actually fit in, and then show intermolecular interactions. They are the kind of glue, glue that by which these molecules can stick onto the protein. So that is called molecular docking. The other one is pharmacophore. Pharmacophore is three-dimensional structural features which are responsible for the biological activity. And third one is protein modeling. I think you guys are aware of this AlphaFold thing that has recently come up. So, but traditionally protein modeling used to be one of the a key component of computer drug design, where the protein 3D structure is not available, where experimentally either using NMR or by using XRD, the structure is not available. They're all by using some template. We used to develop protein model. And then once your protein model is generated, again do a virtual high throughput docking or so and so forth to identify new molecules. Or use the same protein to generate novel structures. They are called de novo. So the other aspect is quantitative structure activity relationship, where you try to derive a this is a kind of a prelude to the data science that we are currently seeing in every aspect of the technology or science. But quantitative structure activity relationship used to be like analogous a series of analogous structures and their biological activity, and application of different statistical methods to derive a correlation between the biological activity and different physicochemical properties. And last one is chemoinformatics, where you try to actually find chemical similarity or structural similarity, and try to find novel chemical structures from the vast possible databases that are actually available. So for each of these things, I can share with you a couple of instances. Of course, they are not anywhere captured. So I'm sharing with you some of the interesting observations. So molecular docking, probably you might have gone through some of the publications, or maybe you might have read in your course curriculum also. So this docking, as I said, this usually the rigid conformation of the protein versus flexible small molecules are fit into a particular pocket by something called electrostatics. So these electrostatics and hydrogen bonds, including hydrophobic interactions, actually determine their affinity to the protein of interest. And there are many success examples, like even if I have to share, ponatinib, which is a CABL inhibitor, has been identified using molecular docking. So and there are at least I remember a vast majority of anti-HIVs, be it protease inhibitors or be it antiretroviral inhibitors. There are multiple drugs which have been discovered using molecular docking. And pharmacophore, of course, cell gelions and some of those things have been discovered using pharmacophore, but pharmacophore per se is not expected to give you the drug molecule. In fact, to tell you very frankly, none of these methods are actually going to give you the drug molecule. They will actually, reduce or they will, they help you to funnel out, vast majority of chemical universe that is there to a smaller number of molecules, and from there you can actually prioritize what is important and then what is, less priority and what is more priority and all that. So that way, pharmacophore is also one of the methodology. But yes, fortunately, we have few examples in molecular docking and protein modeling. Of course, if you look at angiotensin converting enzyme inhibitors like captopril, lisinopril, and all that, enalapril, all that. I think the first generation inhibitor is captopril. So I think using protein modeling, initial experiments, they have actually come to a proline derivative. From there, they have actually found out this captopril, and once captopril is on the market, people developed many co-crystal structures and try to understand their structural biology, and then they have developed other analogs of this ACE, angiotensin converting enzyme inhibitors. Quantitative structure activity relationships, at least the literature says that norfloxacin is one such example where using analogs of 5-fluoroaminophenolines or quinolones, they have tried to identify this norfloxacin, which is an antibiotic. And chemoinformatics, I can cite my own example, like when we were actually working on for the compliance of intellectual property. I'm not taking the names of few of the targets, but try to understand my situation, and I'll try to give as much of information as possible without actually naming the target. So we were working on a nuclear hormone receptor, and nuclear hormone receptor typically has a N-terminal domain, you have a DNA binding domain, and you have a ligand binding domain. This is a typical arrangement of all the proteins. Now your usual nuclear hormone receptors are mineralocorticoid receptor or glucocorticoid receptors. There are multiple, there are many steroid and non-steroidal nuclear hormone receptors. So be it any nuclear hormone receptor, ultimately when ligand is bound, the conformation of the protein changes and the DNA binding domain goes and binds with the DNA, and it induces either transcription or reduces the transcription, and then hence you actually see the biological effects what you see finally. Now, what is important in this case is that this DNA binding domain. So, what actually also happens is that there are multiple mutations upon, say for example, you have multiple drugs which are already there for ligand binding domain. Okay, so you keep treating patients with this ligand binding domain inhibitors or activators. Over a period of time, what will happen is that the ligand binding domain actually develops multiple mutations, and hence it will no more be effective once you start giving the same drug again and again, because of this single point amino acid mutation. The key interaction that is responsible for the molecule or your agonist or antagonist sticking to your nuclear hormone receptor is no more sticking, hence it will actually lose that interaction and it is no more effective. But if you can actually block the DNA binding domain interacting with your DNA, then that's a very novel way of looking at it. And in this respect, I think we tried to do something where there was already a report where the molecule was reported to actually bind at the interface of both nuclear hormone receptor DNA binding domain and the DNA itself. Now, what we did was we have tried to look at different chemoinformatics methodologies to understand the already known ligand and how many different types of molecules are already reported in the literature. And we have tried to identify novel molecules which are extended in multiple directions, and then we have taken all of them using chemoinformatics. We have sifted, and then they are already known in the public domain. So we have taken all of those things, and then we have docked using again I'm going to molecular docking. We have docked at all of those molecules on the nuclear hormone receptor, and we found that certain substitutions at a certain position of already known molecule are showing more potency than the current ligand which is already there in the reported literature. So that's how again chemoinformatics can be used. So proceeding further, I think this is general modeling simulation, modeling and simulation pipeline, where different computational methodologies are used in different stages. Regarding target identification, of course, bioinformatics is very much useful. I think there are multiple targets that have been discovered using bioinformatics, like ghrelin OSL transferase. At least I remember this one to be discovered exclusively by using bioinformatics. And reverse docking, you try to find out the selectivity and all that also using docking. Protein structure prediction we have discussed in the previous slide. So this all comes under target identification. Target validation, target druggability. So how deep is the pocket, whether it is a shallow pocket or a deep pocket, whether this active site where actually the ligand or the substrate binds, whether that is accessible for the small molecule or not. So these all comes under the target druggability. Also, there are multiple algorithms which can actually measure the volume of the pocket. So you need to have some specific volume so that you can actually design molecules to actually fill up that volume within the pocket and hence show the biological activity. And tool compound design, of course, this is a very important to understand the biology of the target itself. And lead discovery, library design, that is high throughput virtual library generation is what library design is all about. Like like combinatorial chemistry, you can also computationally do library design. Docking scoring, that is scoring of say for example you have 100 molecules you have docked, but if you wanted to score which one is on the top and which one is the second pairet and third pairet and so on and so forth. So those can be done by using something called as scoring functions. There are multiple scoring functions which are available. Each one follows a different methodology. Some uses exclusive electrostatics, some uses solvation along with electrostatics, some doesn't use solvation. There are multiple ways how scoring is being carried out. So that is what is docking and scoring. De novo design is that when you have the ligand structure and you don't have the small molecule structure, then what you actually do is that try to identify different pockets within the receptor and try to identify the properties of that pocket. Maybe that particular pocket is surrounded by your hydrophobic residues, hydrophobic slash aromatic. If they are aromatic amino acids which are there, like tryptophan or tyrosine or phenylalanine, then you ought to put something which is again or aromatic, so that there can be a stacking interaction. If that is a hydrophobic like valine or isoleucine and so on and so forth, then you have to actually put something which is hydrophobic there. So there is a key difference there between aromatic and hydrophobic. Although aromatic is also hydrophobic, but there is subtle difference there. So if there is another pocket which is mostly hydrophobic, which are surrounded by threonines or serenins or so on and so forth, then you have to actually put something which is either hydrogen bond donor or acceptor which is suitable for that particular pocket, and then try to stitch these different fragments and then grow it into a complete molecule. Pharmacophore, a moment back we have discussed target flexibility. Yeah, at least a decade back also, target flexibility was a big issue. When I have joined industry, target flexibility was it is over the horizon, that it is very difficult to actually target, because the molecular dynamics simulations which have come into action now. I did my entire PhD on molecular dynamics, and then it took almost like 2 to 3 years only to complete my molecular dynamics simulations. That used to be the scenario when I have started in university. But then now it is like as again going back to examples that Amit gave, like you can actually scale up as number of CPUs and GPUs that you wanted on a cloud, or you can actually Indian cloud service providers are there. There are multiple service providers where you can actually fire your job and then get it done within few hours, which otherwise would have taken a few days or weeks. So because of that hardware and software scalability, now target flexibility is not a very big concern like it used to be a decade back. Now ligand optimization, quantitative structure activity relationship, this is purely a 2D QSAR. And then there is something called 3D QSR where you try to find out three-dimensional aspects of these small molecules, be it hydrophobic interactions. Again, by using something called a methyl probe, you try to understand the hydrophobic interactions. If you want to understand the three-dimensional aspects of the analogous molecules, then you try to put a proton, which actually represents for a positive charge, or some other probes for a negative charge, and try to understand the electrostatic behavior of your molecules. And once you have all these properties calculated, they can all become your descriptors, which can subsequently get you to an equation which will actually predict you what is contributing to your biological activity, and what is positively in fact contributing to biological activity, and what is negatively contributing to biological activity. Structure based optimization, where you have structure of your target available, there you can do structure based optimization. And preclinical tests like in silico absorption, distribution, metabolism, elimination, toxicity predictions. Now of late there are so many softwares which can do this. And physiologically based pharmacokinetic simulations, where actually biochemistry. I heard somebody asking this question. If you want to do actually biochemistry using some of the computational methodologies, how you can do that? You can actually find out what is its km, Vmax, and so in other kinetic parameters, and try to simulate using partial differential equations. And this is something generally called as computational systems balance. So this is the pipeline of computational methodologies that currently, there could be many other applications, but this is in general that I'm talking about, which is apt for for student community to understand what CAD is all about and how pharma industry uses different CAD technologies. Okay, now let me briefly take you through what is rational drug discovery. Rational drug discovery can be put into two different perspectives. One is when receptor structure is known or unknown, or when ligand structure is known or unknown. Okay, so these are the two different larger perspectives within which the entire rational drug discovery goes. Now, when receptor structure is known and ligand structure is unknown, as I said, the three-dimensional structure of the receptor either determined by NMR or XRT, then you do something called as de novo, because now you have protein structure. You can identify different pockets within the protein, and then try to understand the properties of the protein pockets, and then try to build create fragments which are favorable in those pockets, and try to stitch all these fragments and to create a new molecule which is hitherto unexisting. So that's why we call it as de novo. So now the other aspect is that when receptor structure is known but ligand structure is also known, so then we do something called as docking. As we have discussed a moment back, you try to put into the pocket of the protein, and then try to see the electrostatic and hydrophobic interaction, so on and so forth. And hence free energy calculations are also possible. If the co-crystal structure with the ligand is actually available, then you can actually measure experimentally by using something called as isothermal titration calorimetry. You can identify the free energy of binding, and once those kind of experimental results are there, then you can change a small change at a time, and then do something called as free energy calculations. These used to be an academic exercise a decade back. Now I think there are reasonably good softwares which are able to predict binding free energies and free energy perturbations. So that is when ligand structure is also known, as well as receptor structure is also known. And when the receptor structure is unknown and ligand structure is also unknown. Now this is useful. High throughput screening and combinatorial chemistry is useful when in case of infectious diseases, like we have gone through all this COVID pandemic and other things. So when you have the culture of a microbe, and then if you want to see what kind of scaffolds do actually work, then your CombiChem has a library of different scaffolds which you can actually do a high throughput screening to identify the a particular scaffold which is showing effect on the growth of these cultures. So that is when receptor structure is also known, when ligand structure is also known. From there you try to identify the small molecule, and hence from there you can do the analogs of those small molecules to identify or to improve the potency of the cultures of interest. And when receptor structure is unknown but ligand structures are known, Again, this used to be the case. Even now, this is relevant, but it used to be the most often the case a decade back again, where usually, the the XRD technology was not so advanced. Now you have robotics, and you have so many different automations that have come into co-crystallization or crystallization technology. But when it actually started, when the initial structures of GPCRs, all that have come, it was not a very advanced technology at that point of time. Then people used to rely on making different molecules, screening on the on different biological assays, and try to derive the activities, and from activities to structure they used to correlate. And that's what statistical correlation, when it actually comes into picture, then it is called the quantitative structure activity relationships. And pharmacophore, again, using pharmacophore, you also can have kind of structure activity relationship that can be done. So that's what this pharmacophore is doing in this. So molecular docking, I have already mentioned, protein binding site, and then complex interactions with the protein. Pharmacophore, so each of these is a center of the molecule, like protonated amine versus aromatic group, hydrogen bond acceptor, and then this is a hydrophobic region that is there here, and hydrogen bond acceptor within a molecule. So biological activity is a function of these distances. As long as you maintain the distance of 5.2 angstroms between protonated amine and an aromatic group, then you get the biological activity. Similarly, all these things are supposed to be maintained. So as long as you maintain those distances between different features in a molecule, you are bound to get the pharmacological activity or biological activity. That's what is pharmacophore. So this is how the pharmacophore looks like. So this is, traditionally this is ligand based. Of late, there are softwares which people have developed based on the structure based pharmacophore. So now you have ligand based and structure receptor based, and then you develop a pharmacophore. So based on hydrogen bond acceptors and donors and hydrophobic and so on and so forth. So you have different distances that are already here. So based on these three-dimensional spheres that I'm showing here, so these can be given as a virtual screening query to different chemical databases, and those molecules that actually fit these things are selected, and those selected molecules are either purchased or chemically made, and these are biologically tested. Okay, so this is how pharmacophore generation screening actually goes. So QSR, we have already discussed. Based on already existing data, we generally do QSR. So I think how much time we have, 20 more minutes. Okay, so chemoinformatics, again, as I said, based on data collected from the literature, we have raw data pool that is collected both from data conversion as well as collected data literature. Based on this, you actually try to calculate different ADMET database. So why we are doing this is that we are doing all these things to get drug-like molecules. What is a drug molecule? What is a chemical molecule? A chemical molecule may or may not necessarily have toxicity information, but a drug molecule need to have toxicity information. And whether these molecules are orally bioavailable or not, whether the metabolism of this is already reported or not. If it is not reported, did you study the metabolism or not? So if there are novel structures that are coming up, then we have to study all these things. If you have already selected a scaffold which is already reported elsewhere in the literature, then based on already existing physicochemical properties, because usually I'm sure you might have heard about this Lipinski rule of 5 for oral bioavailability. So where hydrogen bond donors and hydrogen bond acceptors should not be greater than 5 or less than 5. So all the properties like log P should not be greater than 5, and then molecular weight of the compound should not be greater than 500. So these are some of the multiples of 5. Or there are 4 properties which are all multiples of 5. Hence, it's called rule of 5. So these properties are calculated for thousands and thousands of molecules, and from there, based on already known drugs and their properties, we can shortlist molecules which are actually complying to Lipinski rule of 5, and then take those molecules and do substructure or a structural similarity search to identify something which is actually having the overall structural similarity with your molecule, but at the same time it has got other extensions or other atomic substitutions. So these all things can be done in chemoinformatics. I'm trying to generalize to a greater extent so that everybody of you can understand. So some of the structural properties and pK being, molecular weight, hydrogen bonds, polar surface area, pKa, lipophilicity, shape and reactivity. Other physicochemical properties like solubility, chemical stability, permeability, and pharmacokinetics and toxicity properties being clearance, half-life, bioavailability, and LD50. So some of these properties can be calculated using computational tools, and these properties can be used for filtering the compounds, or you can actually use them to prioritize compounds for further making and testing. And the other applications in chemistry are in silico toxicity predictions, biocatalysis. I'm sure you might have heard about this. That is using bugs to actually catalyze a particular reaction, or if you identify a enzyme from a bug, then and you see a particular reaction that is actually happening in that particular enzyme, you try to catalyze, you try to mutate it to improve your conversion rate, and hence you can actually do a green chemistry kind of thing. So that is what is biocatalysis, which is a green process. At the same time, this is reusable, and the cost of this initially is high, but then over a period of time this is less. Polymorph prediction is polymorphs are the molecules, the molecular packing in different conditions. So same molecule can pack differently in different conditions when you change the solvent, or when you change the crystallization conditions. And why polymorph is important? Because change in the polymorph changes your bioavailability and other physicochemical properties. That's the reason why polymorph prediction is very important. And at least these three, which are related to process research, are also can be addressed by using some of the computational tools. At least we regularly support our API industry by providing them in silico toxicity predictions, by identifying the genotoxicity, mutagenicity potential of the molecules. And the other thing is biocatalysis. I have also fortunate enough to have worked in few projects in biocatalysis, where we have our few of our colleagues from biotech department. They were actually trying to synthesize the side chain of a drug. So that side chain actually, synthetically it is very expensive. So they were trying to actually identify a bug to do this, and then they eventually identified the enzyme which is actually doing it. But unfortunately, the conversion rate, enantiomeric excess conversion rate that they got was only to the extent of 16%, which is actually commercially unviable. So later, they tried to do different random mutagenesis, and then they could not actually improve it further. So after some time, when they have actually come to us, we have tried to do how this actually the side chain of that particular drug is actually binding, and where from this proton attack is actually happening. Try to understand with some help from our process research chemists, and we try to do some initial mutagenesis in silico mutation analysis, and did docking, and finally molecular dynamic simulations to understand whether this particular side chain is able to retain in the active site of this particular protein over a period of time, or it is immediately thrown out. So some of those parameters we tried, and in fact, one of the first round of mutations that we have generated has given a conversion of 60%. Right from 16%, within first round of mutations, we have given 5 mutations to be incorporated into the protein and in the active site. In the very first round one, the catalysis, or the rate of conversion has improved from 16 to 60%. So that's a very good experience that I have had with my catalysis. Computational toxicology, usually you have many free softwares which are already there. There are commercial softwares which are already there, like Toxday is one of those free softwares which you can use. And this software can be, the results that are coming out of this software, even regulatory agencies also approve this. They also consider the results that you have generated for this APIs for further submission for DMF. So that is computational toxicology, and this is also useful to understand the developmental toxicities related to HERG, cardiotoxicity. I'm showing here. So you can actually develop. There are in public domain, there are so many molecules that are known for HERG binding. So you can take all those molecules. You can actually develop a pharmacophore model. You can develop a QSAR model, or you can use some of the machine learning methods that are reported in the literature to understand the molecule which is potentially new, a drug like molecule. You can actually pass through this database, and then you can actually identify whether this has your molecule has any hERG binding or not, so and so forth. Similarly, you can also do for hepatotoxicity. At least for these two toxicities, we have internally generated predictive models using machine learning methods, and it has been quite successful in identifying. What also usually happens is that once you have these molecules identified, that this has the potential to bind to HERG, which is human ether gogoa gene, which is a ion channel in the cardiomyocytes. So now this molecule goes and binds, and the signaling within the cardiac tissue is blocked, and hence it leads to cardiac related problems. So this is one of the alert that FDA, Federal Drug Administration, Food and Drug Administration USA puts into screening cascade. So we have got several thousands of molecules from the literature, and we have developed a predictive model. And internally, whatever molecules we have passed through this HERG binding model that we have generated, some of them gave to be positive result. That is, they are HERG binders. Now, when we have actually sent it to assay, binding assay, we have seen the binding. So that's how this will help. You will not at least select something which is toxic molecule and send it forward. So those are some of the examples that I can immediately give. And other biology applications of protein modeling, we have said, you might be aware of this AlphaFold. If you are not, please read through Alpha model, AlphaFold, papers that are published in Nature, or what is that company? I forgot the name of the company, but it is from Google. So antibody modeling, currently we are using some of the protein modeling technologies, and then and using loop refinement algorithms, we generate new antibody modeling. Protein aggregation, I'm going to discuss an example with you. This becomes very important in biologics. You have small molecule drugs. Similarly, you also have biologic drugs. So once you develop these biologic drugs and you put into the vial, over a period of time, the attractive forces between these two actually pull each of those protein molecules together, and they aggregate. So in order to reduce those things, either you have to change the excipient or you have to mutate some of the amino acids. So in the first place, to understand whether protein has any propensity to aggregate or not, so this protein aggregation can also be studied using computational methodologies. Pharmacology, yeah, I'll tell you one very good example. In literature, somebody reported that in a particular scaffold was reported to actually cause convulsions in animals. Okay. So when we try to actually see what exactly is the reason for the convulsions, we identified that these molecules have got the propensity to cross the blood-brain barrier, enter into brain. So these kind of molecules to be avoided if your target of interest is in the periphery, not in the CNS. So that way you can actually integrate with pharmacology to identify the the different properties of the molecule, whether they can cross the blood-brain barrier or not. Toxicology, we have already discussed. Pharmacokinetics, based on your log P, polar surface area. Polar surface area is the total polar atoms that are there on a given molecule, and then hence the polar surface area or solvent accessible surface area. So these are all some of the important parameters that comes into pharmacokinetics. And this polar surface area at least is directly related to blood-brain barrier permeation. If polar surface area value is less than 50, then there is a high propensity for this molecule to cross through the brain. Systems biology, again, by using biochemical parameters of the reactions, you can actually simulate a complete pathway. So computationally, that is what you call that as systems biology. Of course, we have done some TNF alpha pathway on this, but then I can't give you any example because for some reason we are not up to what world is doing currently. So protein modeling we have already discussed. Yeah, this is the protein aggregation. This is a Pfizer report. They have reported this in 2007, and after that there are multiple publications. There is a classic example where protein molecule. This is a peptide. There are multiple molecules in the peptide. At t is equal to 0. This is a molecular dynamic simulations, and this time as it extends from 0 to 10 nanosecond to 12 to 40 to 50 to 205 nanoseconds, you see that all the protein molecules have come together and they have aggregated. So that this is how you can actually find out if there is any propensity of your for your molecule to get the aggregates. And predicting new molecular targets for known drugs, I think there are many classic examples. I don't have to tell now, but then you'll know that already approved drugs are used for different indications of late. So the best is during COVID we have used multiple drugs which are predicted to be effective in COVID. So using something called a similarity ensemble approach, reported by Brian Scheuchert and Brian Roth, I think they have come out with this similarity ensemble approach, and using again chemical similarity and structural similarity, some of these exercises can also be carried out. And coming to formulation sciences, liposomes, micelles, nanoparticles, dissolution tests, skin permeability, ophthalmology, that is, eye drops, when you actually put it, most of it actually comes out because of the tear. But if you want actually the more effective drug, then you need some solution which can actually stick to your eye for some time and then slowly permeate inside, and then it can reach to the retina if it is a retinal diseases. So some of those things can be done. We have applied to some extent. And understanding the miscibility and solubility of different polymers. Some of the drugs are highly insoluble, highly hydrophobic drugs. If they are highly hydrophobic, then you need to have a polymer coming into picture, and this polymer should actually dissolve the compound, or it should be able to miscible with the compound, and that should later by different technologies you can make it into as a powder, and then you can make that as a formulation. So some of these things, at least liposomal computational methodologies using molecular dynamic simulations or coarse grain simulations is already reported. Micelles reported. Nanoparticles, we have done that. Over a period of time, we saw that, different excipients in a box of water, how they actually come together and form a nanoparticle. And the size, when we have measured these nanoparticles, they are in the range of, I think, 70 to 100 nanometers range. And when they have taken the TEM or SEM pictures, scanning or tunneling electron microscopy picture, that was more or less in the similar range. So dissolution tests, of course, by using computational fluid dynamics, so CFD, so these dissolution tests can also be done. Skin permeability, again, by using atomistic simulations by molecular dynamics, or using coarse grain methodologies where a group of atoms can be now considered, because atomistic simulations, each atom is considered as an atom. But in coarse grain simulation, what actually happens is that you take a few group of atoms and consider that as a bead. So there are a few notations for it, and using those notations, you can actually make only into fewer beads of a group of atoms. So now this becomes relatively easy for the system to handle. So that way you can actually simulate different bilayers of skin, or different membranes or cell membrane or so and so forth, and you can actually put the molecule, and then you can see that the molecule actually permeating through your membrane to the intracellular side, or through the skin membrane to inside. Ophthalmology, I have just said. Visibility also I have said. There's some of the applications. So this is my last slide. So a few of the benefits of using computational tools are low cost. If you have reasonably good computational infrastructure, then you can actually develop, you can reduce the animal experimentation. In fact, biological experimentation to some extent, because then you have a rationality of why a particular thing has to be developed, why this particular way can be asked, and then some of those things can be addressed. Shorter time to market. If you are actually trying to develop, say for example, I'm just for the sake of example, I'm saying a biology. Now you already have designed a sequence. Now you quickly develop a three-dimensional model and do a aggregation study that moment back I have shown you. If that sequence has aggregation property, then why do you actually want to put money onto that molecule? You better take something which is non-aggregating, but at the same time it has got the similar CDRs and other SDRs intact in that particular antibody. So that way, shorter time to market, or identifying new small molecules from vast, millions of molecules libraries which are available publicly. Development of new products, de novo design can be used for this. Improved competitive edge by using the repurposing, you can actually improve your competitive edge. Or probably, if you already find there are known toxicities for approved drugs, you can actually improve your therapeutic window, and hence a competitive advantage for your molecule will be better. Allows the investigation of sensitivity of a system to small changes in the parameters. Again, this is related to systems biology, where you can actually try to identify the sensitivity of the system. Allowing testing of systems that are costly, dangerous, or time consuming to perform. Say for example, if in chemical industry, if there are particular some reactions which are explosive, and which are highly risky experiments, then you can actually do that computationally. Precise temperature, precise pressure, and all those things can be optimized, and then you can actually take up for synthesis. So that's all I have for today's discussion. I think it's almost getting 5, and for next 10 minutes we can take questions.
Yes, sir. Thank you, sir, for that very interesting session. So, yeah, we'd like to have now questions. So, you can put it in the chat box, or you can just unmute and ask. Already, I think we have a question in the chat box, sir. Can you please take it?
Hello, I think there's a question that has come up. Can you
Yeah,
explain
yeah.
the virtual screening and its role in drug design? Yeah, I think virtual screening. I think I'll actually put in two perspectives in front of you. Earlier, what people used to do is that identify. Once they have a receptor that is identified, then you have many chemical vendors who have made electronic libraries. You can take all those libraries and try to identify molecule which actually fits into your pocket. Number one. There are focused libraries which are also available, like kinase library, GPCR library, and so on and so forth, which again they are also electronically available. If the target of your interest is a kinase, then take kinase library and initially try to find out what kind of structures can actually get into your kinase structure. So that is something related to virtual screening. So let me also tell you, Saurav, that virtual screening is you are actually trying to find out a new molecular structure for a new receptor, or you are trying to find out a new molecular structure for already existing protein structure with existing molecules. Say for example, let me give an example of AC converting, angiotensin converting enzyme inhibitor. Say for example, captopril. There is a problem with the captopril. Now you want to develop a newer molecule for angiotensin converting enzyme protein, which can actually fit more properly with less side effects. Just imagine that captopril has some side effects. You want to reduce that. So how can you do that? You can actually take this captopril molecule, develop a pharmacophore, and screen in millions of molecules database. Try to get a molecule which is having similar pharmacophore but different structure, and then therefore the toxicities or side effects that you have already seen with your earlier molecule are not there in this. Number one. Number two, by using similarity approach, captopril you take, do a t-dimensional similarity search, and then screen across all the databases, again get it, and then do the docking, and then try to identify the intermolecular interactions and hence the potency. And third one is that of late you have the generative algorithms and other these AI methodologies which have come, where using these some of these methodologies you can actually identify the space which was hitherto unexplored, and then you can actually develop novel structures and then dock it and see. So there are these are the different ways that pharma industry actually using computational tools to identify virtual screening. Have I answered you, Saurav?
Yes.
So there is another question, sir. I want to know if pharma industry look for people from human genetics. Those can focus on preclinical trials and the role of genetic variation in drug target mutation and prevalence in population. Of course, there. See, pharma industry only needs people who can give them opportunities. So it's an industry. Obviously, industry is there for their business. They want to make money. So as long as you can give them an idea by using genetics, that using genetics this and this, you can actually identify some of these experiments, like of late this CRISPR Cas9 experiments which have come. You can actually validate them genetically to either knock out a particular gene and try to identify how important is that particular target, and whether knocking out of that target is actually inducing resistance or sensitivity for your drug or not. So all those things are part of genetics, and of course people have already started looking at it, and there is no dearth of applications that from genetics have come into drug discovery. Precision medicine, same as drug discovery, and which is more cost effective, more economically viable. Precision medicine is an advancement in drug discovery. Is a drug discovery is a general term. Any methodology, be it computational, be it experimental, anything you do is a part of, as long as you are discovering drugs, it's drug discovery. Precision medicine is wherever you want to precisely target a particular patient population, then the precision medicine actually comes into picture. Of course, both you can say that part of drug discovery, precision medicine should be part of drug discovery. Is not same as drug discovery. If DNA sequence is changed by mutation, can bioinformatics help to identify and correction? Of course, this is this is doable. This is what usually it is. People do it. What are the qualifications to join a compulsory medical company after MSc? What are the opportunities as a new bioinformatician? What kind of package is there? There is nothing like no qualification. But if you are a bioinformatician, you should know, you should be updated with how different targets are identified, and the applications of drug discovery. I'll just tell you, share an example, a case study with you, since you have asked this question. Of late, in one of the project, we were actually working on a small molecule, and in a cancer setting. We were actually generating molecules based on the receptor information, target information. And in biochemically, it shows a very good binding, it shows very good potency and all that. But the moment we actually go to cell lines, it did not work. So these are all cancer cell lines. None of these molecules have shown good effect on those cancer cell lines. Then, using bioinformatics tools. The reason why I'm sharing you with this is that because of bioinformatician, this new, information had come out. What my bioinformatician did was, he tried to do analyze the transcriptomic information and transcriptomic information that is coming from the cell lines. And now when he has superimposed all those things, and the kind of setting in which I am actually trying to get my molecule tested, and the kind of cell lines that I'm taking are not talking to each other, there needs to be a deletion and a mutation in two different genes in the setting that I am working. These two are not there in the cell lines that I have taken. And how do I expect that these kind of things will actually show up activity? So I think after this information had come up, we have asked like our biology colleagues if they can actually by using different databases, we have tried to identify exact cell lines matching the phenotype of the disease that we are actually working on. Then we have identified from the database sources and said that these are 3 or 4 cell lines that you should be using. So they went back, they identified those cell lines, and the activity started showing up. So that's how bioinformatics can come into picture. So I hope I have answered, Piyush, how bioinformatics help in biotechnology and molecular biology. Yeah, of course, for designing siRNAs and identifying the best antibodies that you could use for your experiments. There are multiple applications. I mean, the only thing is that you should keep updated with various applications that people are actually using it. But per se, in at least with my little experience in pharma industry, bioinformaticians per se, the biology or molecular biology may not actually recruit them. The reason is that they want more experimental hands than bioinformaticians. So actually, they actually come into bioinformatics department where they actually support different department. That's how I have seen in my little experience in different pharma settings. So that's how I would rather put it, like you can actually help them in multiple ways. So in fact, I'll tell you another example. We were actually working on a antibody modeling project, and it was observed that at protein level, the antibody that these guys have generated binds in picomolar potency. But the moment you take it to a cellular level, it doesn't bind. It shows in one cell line, it shows binding, or rather toxicity to the cell. The other cell line doesn't show anything, no cell killing. Then we try to do this interactome analysis using the string database and other things. So we try to understand, taking this particular target of our interest and to which this antibody is actually binding. We have taken this target and tried to identify the complete interactome of this, taking high confident interacting proteins, and then we try to segregate them or filter them based on the extracellular property, or gene ontology terms. Based on gene ontology terms, we try to identify them which are extracellular, which are cell surface expressing proteins, and so forth. And then we have identified that in some cases, when this antibody is not able to bind to this particular receptor of our interest, in fact there is another partner which is actually binding to this receptor. So that's another information that we found while we were doing this. I think these are the 2 examples that I could immediately give you for bioinformatics. Muskan asks, how can I directly use any software for molecular docking by using protein data bank? You can use Autodoc, which is freely available, and download proteins from protein data bank, and prepare them and use it in Autodoc, and then take a small molecule library. Again, they are also available freely. You have zinc database which is freely available. Take it and do docking. Saurav, another question. Like every time mutation occurs in SARS-CoV-2, so how we come from this in case of drug discovery? As long as you have an algorithm which can actually predict the next set of mutations, which should have high confidence biologically. So I think once that kind of confidence is there, I think you can again go. See, you say any number of technologies, but it is important for any regulator to approve. At least during first wave of COVID, there was no other option, so there was emergency approvals that were given. I don't think so. Going forward, we have already some repertoire where people can use it. Some medicines are already there; they can use it to some extent. If you want to precisely do that, I think there is this drug which Pfizer has come out with, Praxavate something. It's a very complicated molecule. So they have already come out with that molecule. But I'm sure there are so many drugs that can be discovered. But again, you can't as long as you can't predict the next biologically important mutation, you cannot do anything there. Can I use bioinformatics in experiments related to restriction endonuclease? Maybe, may not be. I cannot answer you, Avra. Okay. Okay. I think there are no more questions.
Uh,
So,
if some participants have any
Suvarna or
other question, they
Dr.
can
John,
unmute and ask.
the first skill that you need to develop is try to have that imaginary picture, like what exactly happens, right from target identification, target validation, and then trying to identify the assay methodologies. You should be very well versed with any of these things, almost all of these things. You should be knowing about discovery biology, biology of the disease especially, and different assay methods like fluorescence based assays or binding assays or ELISA based assays, or CRISPR Cas9 methodologies. So some of these things, at least theoretically, you should be knowing what exactly happens in some of these things. At least I'm assuming that all of you are coming from biology background, hence I'm only sharing the skills to be developed in those areas. So some of these things are very important. Disease biology is absolutely important. Please keep yourself updated about the disease biology latest therapeutic modalities, like whether be it a small molecule, be it antibody, be it there are different types of antibodies again. So antibodies, be it ASO or saRNA, so and so forth. You should read up all these things, and then that way you will be able to answer the questions that scientists may ask you. So this is a brief skill set that I have for for people who are coming from biology background, but chemistry background it may be quite different. I'm assuming that all your students are from biology background.
Okay. So thank you, sir. Thank you for that lovely session, and that was very interesting one. And with this, so we have come to the end of today's webinar. For the participants, I would like to share one thing, that there is a feedback form link that has been shared with you in the chat box. So you are requested to fill up the feedback form, and based on that you will be provided your e-certificate. So I request all of you to fill up the feedback form. It's there in the chat box. Please check. And Professor and Dr. John sir, I would like you to take the opportunity and give the vote of thanks, sir.
Yeah, okay. Yeah. So thank you, madam. I hope I'm audible.
Yes, sir.
Yeah. So good evening to all. I sincerely thank Honorable Vice Chancellor Dr. Subir Bhattacharya for his constant support. The next, I'd like to thank Registrar Professor Pranav Ghosh and Dean Faculty of Science Professor Subhash Chandra Rai for their motivation and encouragement. Also, I thank the finance officers and other university authorities for the motivation of this event. I thank our eminent speakers, Dr. Amit Das and Sairam, for their inspiring speeches. Sir, both of your talks were wonderful and realistic about the latest trend in the field. Really, you are given a good talk. Thank you very much for that. I also thank my dear colleagues, Dr. Subarna Thakar and Dr. Chiranjeev Sarkar for their for this events plan and executions. Without them, it is not possible to get success. I thank all research scholars, students of our department who helped us with the success of this event. Last, I would like to thank all the enthusiastic participants who patiently listened to the talk till the end. Thank you for your kind support. I hope we will meet at another event soon. Thank you once again.